being the structure and organization of actin filament. Actin filament is a major cytoskeleton protein of most cells in which it polymerizes into a thin filament. This filament is approximately around 7 nanometer in diameter and several micrometers in length. These actin filaments are also called as microfilament. The assembly and disassembly of actin filaments, their cross-linking into bundles and networks, and their association with other cellular structures are regulated by a variety of actin binding protein. And these regulations also leads to the flexibility of the cell movement, cell motility and mobility. So the assembly of actin filaments and disassembly of actin filaments we will be seeing. The three-dimensional structure of both individual actin molecules and actin filaments were determined in around 1990 by Kenneth and their colleagues. And each actin monomer, we call it as globular actin molecule, which are tightly bound head to tail interaction so that the actin polymerizes automatically to form a filamentous F actin. The globulous actin protein's molecular weight is around 375 amino acid of 43 kilodalton. So all the actin molecules can assemble. So these assembly will require ATP molecule. So automatically the G actin, which is a monomer actin, which will first bind to ATP molecule. So once it is bound to ATP molecule, the G actin, which is bound to ATP molecule, can only form the dimer, then a trimer, then a filamentous actin. So in the way of formation of filament, the nucleation of the filament will be taking place. So once the nucleation of the filament is taking place, so two ends will be formed for the actin molecule. Since it is very polar in nature, we get plus end and minus end. The plus end of the actin and the minus end of the actin, both assembly and disassembly of actin will be taking place. In the plus end, the G actin, which is ATP bound, will go and add, and in the minus end, the ADP actin will be released from the minus end. So in both the end, polymerization as well as depolymerization will be taking place. The difference is in the plus end, the rate of polymerization will be more when compared to rate of depolymerization. Whereas in case of minus end, the rate of depolymerization will be more when compared to rate of polymerization. So that makes the growth of the plus end and the decrease in the rate, the decrease in the length of the minus end. So in the plus end, we can see more the G actin plus ATP conformation. Whereas in case of minus end, G actin with ADP conformation will be seen. So ATP actin will be polymerized and ADP actin will be depolymerized. We have in the nucleation step two important proteins. One is profilin. This profilin is used to convert your ADP actin to ATP actin molecule. So for the initiation of the polymerization, formin dimer will be used. So this formin dimer will first start the binding of one G actin, followed by 2 as a dimer, then the third molecule as trimer. So once if the trimer is formed, the further polymerization will be taking place so that plus end and minus end of the protein will be formed. So the new branches of this filamentous actin. So branching will be taking place with a special protein called ARP2/3 protein. So this ARP3/2 protein will go and bind to the plus end of the actin filament, which is shown in the yellow color. Once if it is bound, furthermore addition will be branched. So automatically the branching will be starting. The new branching of the filament will be done with the help of ARP2/3 protein. And we have something to stabilize this entire actin filament. For stabilizing the entire actin filament, we have special proteins, we call it as capping protein. So we have two different types of capping protein. One is plus end capping protein, the other one is minus end capping protein. Plus end capping protein goes and binds to the plus end and it protects the plus end. Whereas minus end capping protein goes and binds to the minus end and it protects the minus end. So we have another stabilizing protein which can stabilize the entire filament. So for stabilizing the entire filament, tropomyosin will be present, and the tropomyosin will cover the entire filament. So the entire filament will be stabilized and protected with the help of tropomyosin. So both positive plus N capping protein, minus N capping protein and tropomyosin will increase the stability of actin filament. So the stable actin filament can be assembled into two ways. One is actin bundle, the other one is actin network. As actin bundle, it will be arranged parallelly and the cross-linking protein will be present. So parallel bundles will be formed. And in case of networking, we have cross-linking of these actin filaments, and the cross-linking protein will randomly cross-link these networks. So your actin filament, which can be assembled into both bundle as well as network. So along with that, we have another different protein called cofilin, which we call it as serving protein. So this serving protein will be used to cleave the actin filament. In order to form either cross-linkage or bundle, we need different kinds of length and different structures of actin filament. In order to do so, we have another serving protein which is cofilin. This goes and binds to the middle of the actin filament. So once if it binds to the middle of the actin filament, it breaks the actin filament into two. So once if it is broken, a new filament will be formed. So one single filament can be contributed as two different filaments, and new plus N and minus N for two filaments will be formed. Automatically, these kinds of broken actin filaments can be added either to the bundles or to the networks. This is how the actin bundles and networks are formed, and these will be served below the plasma membrane of almost all the cells. Thank you.